we're going to start by talking a little bit about the expectations for this course, and then going into some of the basics of molecular biology and talking a bit about the history of the field to get us off on the right foot for this semester. So let's start by talking a little bit about the expectations for this course. We're all in a little bit of a weird place right now as we're living through this pandemic and these very odd times, and classroom formats are changing as well as the methods for which people are going through and teaching these different classes. For me, this is my first time teaching molecular biology to engineers, and it's also my first time teaching online. So I ask that you bear with me as I navigate some of these changes, and be patient as we try to work the kinks out in this course together. And if you have any questions or any problems, please let me know immediately, and let's just make sure we stay in really good communication to be able to navigate these murky and uncertain times together. So let's start by talking about expectations. My goal as an instructor is to try to have these lecture videos prepared, edited, and posted for you on YouTube by at least Monday night of every week. That gives you a full week to go through, watch the lectures, make sure that you do the readings so that you can do your highlights assignments that we'll talk about here in just a second. So that's the first expectation, and that's for me. I am going to try very hard to get things posted by Monday night. I'm not going to guarantee that that's going to happen every week, but that is going to be my goal. And whenever I have a lecture finished, what I will do is post it on YouTube with an unlisted link, and I'll send everybody in the class an email that has that link so that you can go through and watch the video. And if at any point there's any issues with the videos or things are missing or skipped, please bring it to my attention as soon as you see it, because if I posted it, I didn't know that there was an issue with it. The second expectation is for you. So at the end of every week, when you've had time to watch my lectures and do the readings, maybe watch some of the supplementary videos that I'll be sharing with you throughout the semester, that's when you're going to put together your assignment that's a weekly assignment known as your highlights. And it talks a little bit about this in the syllabus. I talked a tiny bit about this in the introductory video. So what you're going to be doing is sort of summarizing and synthesizing all of those things that you learned from the readings and from watching the lecture videos, and maybe from those supplementary videos too if you'd like to include that, though that part is not required. And just summarizing those main concepts, the takeaways, the final most important points of those different lectures in your mind, and those readings in your mind, into these one-page assignments that are just little bullet points. You can organize them into sort of an outline format if you want to. That I will leave that up to you. The only requirement that I have for that is that you not use Calibri, because it drives me crazy. I don't care what font you do use. I prefer Times New Roman. If that's not your jam, that's fine, but please don't use Calibri. It just makes my heart sad. So that will be your assignment every week. That will be due by the end of the day Friday. And when I say end of day, I'm talking like end of the business day. So by 5 o'clock Friday, I better have your highlights pages in my email inbox. So please make sure you get those to me. Next is discussion boards. So there are no requirements for posting to discussion boards for this class. However, I have set up discussion boards for each of the 12 weeks prior to exam number 2, and then after exam number 2, we're going to do some different things for the semester. But up to that point, there's going to be lectures and readings, and I would like for you to go in each week, look at some of the questions that I've posed or some of the things that I've asked you to do within that discussion board, and just throw a couple threads in there, maybe give your 2 cents. If you see that a classmate has a question, they can post that question in there and you can go in and give what you think the answer is. Or if you have a question, you can post a question in those discussion boards along those threads. So this, even though this is not a requirement for the class, I do have an expectation that as graduate students, you guys are going to be using this as a way for you to communicate, since we aren't really going to be meeting in a face-to-face format or in a synchronous format via Zoom or anything like that. So this is all your class engagement. And I will be monitoring those boards, and some of that will contribute to your overall participation grade for the semester. So don't blow it off. There's not a requirement for how many times you need to post or anything like that, or the kinds of things you need to post. I just expect you to be engaged in the discussion on the boards. And then the last thing I wanted to tell you about the class is that the details for exams, exam 1, exam 2, and the final exam, which will be comprehensive, and also the project that you're going to be doing later on. I'm still working on some of the logistics and the details for that, so I'll be sharing that information with you in the coming weeks as we work out some of those details. Okay, so now let's actually get into molecular biology. What is it? Why do we care about it? Where did it come from? We will explore all of these questions and more in the coming minutes. Okay, so let's dive right in and just define this term of molecular biology. When we say molecular biology, what we're talking about is this series of interactions that are happening on the molecular level, both within cells themselves and between cells that make up the tissues of an organism. This field of biology examines the underpinnings of things like replication, transcription, translation, and cellular function. This field often overlaps with the fields of biochemistry and genetics. And when you start looking into the literature, you might find references to something known as the central dogma of molecular biology. But this central dogma of molecular biology can really be extended to be the central dogma for all things related to biology. When we talk about the central dogma of molecular biology, what we're referring to is how genetic information flows into protein synthesis through the processes of transcription and translation, two things that we're going to spend a little bit more time talking about in the coming weeks of this course. Some of the major players in the molecular processes that we're going to be discussing are DNA, RNA, and proteins. You might recall that DNA and RNA are comprised of nucleic acids, while proteins are comprised of amino acids. DNA and RNA are intimately involved in the design and construction of these different proteins that we use for all different types of biotechnologies, as you'll learn later in the semester. The term molecular biology wasn't coined until the 1930s, even though many of the biotechnologies that we're going to be discussing later in the semester were developed long before that term molecular biology became an official part of the scientific lexicon. The field was described beautifully in a 1961 edition of Nature by a fellow named William Astbury, and this is what he had to say about molecular biology. Not so much a technique as an approach from the viewpoint of the so-called basic sciences, with the leading idea of searching below the large-scale manifestations of classical biology for the corresponding molecular plan. It is concerned particularly with the forms of biological molecules. And we're going to talk a lot about biomolecules and how they play a role in this area of study. Some of the earliest work regarding our understanding of the structure of DNA dates back all the way to the 1860s with this guy, Friedrich Miescher. And I probably butchered that name, but that's the best I've got right now. Now, what this researcher did to make his mark on human history was discover something that he called a nucleon. Today, we refer to this substance that he discovered as nucleic acids, and they play a pretty major role in molecular biology, as you'll see over the coming weeks. And this is how this story goes. He was examining pus-filled bandages that he got from a colleague at a local clinic, and what he was doing was looking for various types of proteins that are found within leukocytes, or the white blood cells of the human body. As he started to process some of these different leukocytes and filter them out and try to get at the meat of what some of these different proteins were, he accidentally discovered this substance that didn't behave the way that proteins normally behave. And he started characterizing what was different about this substance, and eventually termed this the nucleon, which would then go on to become what we know today as nucleic acids. Some of the things that made this substance so unique was that unlike other proteins, it couldn't be broken down by proteolytic or protein digesting types of enzymes. It also had a higher phosphorus content than what proteins have. When he wrote about this, this is what he said. It seems probable to me that the whole family of such slightly varying phosphorus-containing substances will appear as a group of nucleons equivalent to proteins. While through time his name sort of fell into obscurity and later got sort of rediscovered by people really interested in the history of science, other scientists were starting to investigate the chemical compositions of some of these little substances, these nucleons that he had discovered. One of the many researchers involved in this kind of work was Thebbius Levin. He was a physician who became a scientist and wrote over 700 papers that made their way into the scientific literature. He was the first person to report the three major components of a single nucleotide: the phosphorus, the sugar, and the base. He also discovered and described ribose, which is a carbohydrate that makes up the R in the RNA, as well as the deoxyribose, which makes up the D in DNA. Additionally, he was the first person to talk about how DNA and RNA molecules were able to fit together. His groundbreaking work was later expanded upon by this guy, Erwin Chargaff. Now, Chargaff was the first person to report that the composition of DNA varies from one species to another. So we're seeing some real differences at the organismal level based on these molecular components. He was also the first person to note that all DNA, regardless of species, maintains certain properties, such as the amount of adenine, or A, being similar to the amount of thymine, or T, as well as the amount of guanine, or G, being similar to the amount of cytosine, or C. These are going to be things that we talk about in more detail in coming lectures, so you'll be getting really familiar with those letters. This phenomenon of the relative equivalencies between these different components of DNA became known as Chargaff's rule, and it's something that we'll return to a little bit later in the semester. Okay, so later X-ray crystallography work that was conducted by Rosalind Franklin and Maurice Wilkins would prove to be super important in trying to understand the structure of DNA. In 1951, Rosalind Franklin delivered a lecture in which she talked about the two different types of forms of the DNA molecules, type A and type B. In this lecture, she also talked about how phosphates form the external structure of DNA and a little bit about how water played a role in helping to maintain the stability of the DNA molecule. This lecture was attended by one James Watson, who would go on to work with Francis Crick in being able to describe and create the model of the DNA double helix structure. And much of their understanding of this model came from the work that was done by Rosalind Franklin, even though she didn't really get credit for it. At one point in time, she was working on taking these X-ray photographs of bDNA. And one of the famous photographs from those time periods when she was working was a photograph known as photograph 51. Photo 51 is at the center of a scientific controversy regarding the intellectual property rights between Rosalind Franklin and James Watson as to who it was that really had the right to talk about the overall structure of DNA and get published scientific recognition of those results. By 1952, Franklin and her student Gosling had significantly redefined the critical parameters for building the model of the molecular structure of DNA as we know it. This work was used by Watson and Crick when they were developing their own model of DNA, which they published in 1953. When they published that research, they gave no credit to Franklin or her student Gosling for the critical groundwork that was laid by their research. Over the next couple of weeks, we're going to go into a lot more detail about the structure and the composition of both DNA and RNA. Following the DNA work of the 1950s, molecular biology as a discipline began to pick up momentum and grew into its own in the 1960s. New discoveries in the fields of biochemistry during that time period, such as the demonstration of in vitro protein translation, the demonstration of genetic codons, and the earliest principles of polymerase chain reaction, or PCR, began to emerge in this decade. PCR is something that was first proposed in this decade by a couple of researchers whose names I can't pronounce, but I'm probably going to put them in the video somewhere in here. Subsequent work by various researchers in the 1970s would lead to the development of things like recombinant DNA technologies, cloning, and genetic sequencing techniques. By 1982, the FDA had approved the very first recombinant protein drug, insulin, and this was approved for the treatment of human diabetes. This may seem like a small thing to us now, but up until that point, people were getting insulin from extracts taken from cows and pigs, and there were a number of side effects that came along with taking those kinds of drugs. In 1987, which, let's just all agree, was the best year, we saw Susumu Tonegawa win a Nobel Prize for the work that was done on understanding how antibodies were produced. Having this understanding of how antibodies produced helped to lay the groundwork for some of the most fundamental things that we use in molecular medicine today in terms of diagnostics for various forms of human diseases. Another important thing that happened that year was that we saw the first criminal case where DNA evidence was used to be able to gain a conviction. 2 years later, Alec Jeffreys developed a technique for using electrophoresis to examine DNA polymorphisms, creating the birth of what we now know as DNA fingerprinting. Now we hop forward into the 1990s, where we see the beginnings of the Human Genome Project, which most of you are probably familiar with. We also start to see the beginnings of gene therapy and the advent of using some of these technologies to be able to treat human disease. We also see the birth of the entire field of bioinformatics as computers really start to take over and big data becomes big business and big news in the scientific community. And finally, towards the end of this decade, we get to meet Dolly, who was a sheep that was cloned by people using molecular biological techniques. Okay, so later in the semester, we're going to focus on molecular biotechnologies. This is a field of research that combines the principles of molecular biology with the principles of engineering, along with a variety of different types of tools that can help us to process materials produced by biological agents in order to provide a variety of goods and services. This field of molecular biotechnologies pulls from a variety of other areas, including microbiology, genetics, biochemistry, immunology, cellular biology, and chemical engineering. Alright, so that's enough of a primer on molecular biology for all of you for right now. If you want to learn just a little bit more about the basics of molecular biology or the history of the field, or maybe just listen to someone who isn't me talk about molecular biology for a little bit, there will be a number of supplementary videos that I'll add into the email whenever I send you the link to this video. Be sure to get your highlights to me by Friday of this week. I know I'm getting this video posted just a little bit later than planned, but I still expect you to have the assignment done by 5 o'clock on Friday. It really shouldn't take you too long because this was a short video and the reading was like less than a page. You can do it. I believe in you. Next week, we're going to be taking a look at the structure and function of DNA. Make sure that you read chapters 2 and 3 in your OpenStax textbook before you watch that lecture. Thank you all for watching, for tuning in, for paying attention, and for getting excited about this semester's class in molecular biology for engineers. We'll see you soon!